Welcome, everyone, to the Deep Dive by RevGenetics. This is the show where we take some really dense, complex research and break it down. We want to give you the key insights so you can stay informed, without getting lost in the jargon. And just a reminder, this deep dive is proudly supported by RevGenetics. They're all about bringing you solid science and health education. You can listen on Spotify or watch us over on YouTube. Today, we're jumping into something truly, mind-blowing. It's a study from 2025, seems recent, right? Where scientists used genetically engineered human stem cells. And get this, they managed to reverse multiple signs of aging in primates.
Yeah, it really does sound like something out of a movie, doesn't
It
it?
absolutely does, but it's real science, a huge step forward. It gives us this glimpse into, maybe redefining aging itself. So let's get right into it.
Okay.
So the core of this involves senescence-resistant cells, SRCs. Now, for those of us following cellular health, but maybe not working in a lab, what exactly are these SRCs? And, what makes them so special in this whole fight against aging?
Great question to start with. Think of your body like a massive community of cells, trillions of them. As we get older, some cells, they kind of enter this state called senescence.
The zombie cells, right. I've had that term.
Exactly. That's a common way to put it. They stop dividing, which might sound okay, but they don't just disappear quietly. Instead, they start pumping out this mix of harmful stuff, inflammatory signals, other damaging molecules.
So they spread the problem.
Precisely. They can damage healthy cells around them, speed up aging systemically. Now, these SRCs are based on a type of adult stem cell human mesenchymal progenitor cells, or MPCs. These MPCs are already pretty useful therapeutically. They don't usually trigger a big immune response.
So less rejection risk.
Right. And they have really good anti-inflammatory properties naturally.
Okay, so they start with something already beneficial, but the key here is senescence-resistant. How did they make them resistant, and why is that specific part, resisting aging themselves, so important?
That's really the clever bit, the genetic engineering. The scientists specifically went in and boosted the activity of a gene called FOXO3.
FOXO3, I think I've heard of that one. Linked to longevity.
That's the one. It's considered a major player, a master regulator for keeping cells resilient and promoting long life, found consistently in human longevity studies. By turning up FOXO3, they essentially gave these SRCs, like a superpower, made them super tough against the kinds of stress that normally make cells senescent.
Okay.
So they last longer in the body, they can keep doing their job fighting those harmful senescent cells without becoming part of the problem themselves.
Giving the good guys shields, essentially.
Kind of, yeah, a built-in defense.
And this wasn't just a quick look. This was a pretty long study, especially for primate research. Can you walk us through the setup?
Absolutely. It was a big one, 44 weeks. That's a serious commitment in primate studies. Shows real rigor.
Definitely.
They used aged cynomolgus monkeys between 19 and 23 years old.
And what's that in human years, roughly?
It translates to about, 57 to 69 years old for humans. So definitely getting up there. The SRCs were given intravenously, just into the bloodstream. And they compared the results carefully against two control groups.
Right. Controls are key.
Yep. One group got just saline, salt water. The other got the original stem cells, the MPCs, but without the FOXO3 boost. They call those wild-type MPCs or WTCs.
So engineered versus original versus nothing makes sense.
Exactly. That comparison really helps isolate the effect of the genetic enhancement.
Okay, critical question time. Safety. Especially with genetically modified cells, what did they find? Was it safe?
That's always a huge concern, and the news here was very good. Excellent safety profile, actually.
It is great to hear.
Throughout the whole 44 weeks, they saw very few adverse health effects. Minimal.
Yeah.
And maybe the most important point, addressing that common worry about cell therapies.
Tumors.
Exactly. None of the animals that got the cells developed any tumors over the entire study period.
Wow, that is significant. Huge hurdle cleared, potentially.
It really is. It adds a lot of confidence moving forward.
Okay, so safety looks good. Let's get to the results that everyone's waiting for. What actually happened to these monkeys systemically? Did they really, get biologically younger? Can you even measure that?
Yes, they did. And yes, you can measure it remarkably well, actually. The researchers used these sophisticated tools called aging clocks.
Aging clocks, how do they work?
Well, they look at patterns across the body that change predictably with age. Biological age, not just birthdays. They use two main types here. One looks at gene activity, the transcriptome, the transcriptage. The other looks at tiny chemical tags on DNA called methylation marks, DNAM.
Okay, two different clocks. Does using both make the results stronger?
Absolutely. It's like getting two independent confirmations. Provides a much more robust picture of genuine biological age change. They even developed a specific reverse score to quantify how much aging was pushed back.
So what were the numbers? How much younger did they get?
The results were, well, pretty astounding. The SRC treatment, the engineered cells, reduced the monkey's transcriptomic age, so based on gene activity, by an average of 3.34 years.
3 1/3 years younger biologically.
Wow, and that was significantly better than the standard NPCs, the WTCs. They only achieved about 2.80 years,
So the engineering made a real difference.
clearly. And it wasn't just one or two spots. About 54%, over half of the tissues they analyzed in the SRC group, showed these age-slowing effects.
Over half the tissues, that's systemic, isn't it? Not just localized.
Exactly. It suggests a widespread recalibration of the body's aging process, like hitting a biological reset button across multiple systems.
Can you give some specific examples? Where was the reversal most dramatic?
Yeah, some tissues showed really remarkable rejuvenation. Skin, for example, a huge reduction. 5.56 years biologically younger. Think about the implications for visible aging.
5.5 years off the skin's age.
Yeah.
Incredible.
Lungs saw about a 4.08-year reduction. Skeletal muscle improved significantly to abdominal muscle by almost 5 years, 4.91. Quadriceps by about 3.5 years.
That could mean better mobility, strength.
Exactly. Huge for quality of life.
Yeah.
Even internal organs like the spleen, about 2.55 years younger. And really importantly, the hippocampus in the brain, vital for memory, showed a 2.03-year reduction.
And did the other clock, the DNA methylation one, agree with this?
It did, which is really compelling. Where the transcriptomic age reversed, the DNA age clock generally showed similar reversals. For instance, the brain showed almost a 5-year reversal, 4.98 years, on the DNA age clock. Skeletal muscle over 4 years, 4.02. Spleen over 2 years, 2.12.
That dual confirmation really strengthens the findings, doesn't it?
Absolutely. High confidence in the age reversal they observed.
So beyond these age clock numbers,
Yeah.
what did things look like, under the microscope, at the tissue level? What actually improved inside the monkeys?
Yeah, the histological level, the tissue level, showed really profound holistic improvements. First off, a significant reduction in those senescent zombie cells we talked about.
Fewer bad apples.
Exactly. Across many organs,
Yeah.
lung, liver, heart, brain, skin, spleen, kidney, pancreas.
Mm.
Pretty much everywhere they looked.
Wow.
But it wasn't just clearing out the bad stuff. They saw restoration of normal cell structure, like the nucleus looking healthier. Chronic inflammation went down.
Yeah.
Less immune cell infiltration, lower levels of inflammatory signals like IL-6 and TNF-alpha.
That inflammaging idea tackling that.
Precisely. That low-grade chronic inflammation drives so much age-related disease, and they saw improvements in age-related damage. Blood vessels looked rejuvenated, signs of neural restoration of the brain, less metabolic dysfunction, and less fibrosis.
Fibrosis, that's like scarring, right? Hardening of tissue.
Yeah, that harmful thickening that stops organs working properly. So reducing that is a big deal. It really paints this picture of comprehensive repair, system-wide rejuvenation.
This is just, it's incredible research. Really highlights how working at that deep cellular level can impact overall health and vitality. And that's actually the kind of philosophy that drives our sponsor, RevGenetics. They focus on ingredients backed by science, things like Fisetin, NMN, metacurcumin 277X. These are compounds aimed at supporting the very things we're discussing: cellular resilience, healthy aging pathways. It's genuinely exciting to see studies like this one confirming the potential of approaches they've believed in for years, really aiming to support your cellular health, offering solutions rooted in science to help you on your health journey. Okay, so back to the monkeys. We have this astounding data on biological age, tissue health, but what about, real-life improvements? Did the monkeys act younger, better function? And what could this all mean for us, for humans, down the road?
Yeah, the functional benefits are really where it gets exciting for potential applications. Cognitively, for brain function, the SRC-treated monkeys showed significantly better memory retention. They used a specific test, the Wisconsin General Test Apparatus Delay Task, basically testing recall after a pause.
So, better short-term memory.
Essentially, yes. And MRI scans back this up. They saw preserved thickness in brain areas like the frontal and parietal lobes areas that usually shrink with age.
Wow.
And better connections, both structural and functional, in key regions like the hippocampus and prefrontal areas. Crucial for working memory, complex thought.
So the brain wasn't just looking younger on scans, it was working better.
That's the key takeaway. Even myelin thinning, that insulation on nerve fibers, was reduced. better signal speed.
Amazing for potential cognitive decline therapies. Did they look even closer, like at specific brain cell types?
They did, using a technique called single-nucleus RNA sequencing on the hippocampus, very detailed. Found that SRCs rejuvenated the various cell types there by an average of about 2.5 years. And some types showed really dramatic effects. Immature inhibitory neurons, almost 7.2 years younger,
7 years.
and immature excitatory neurons, over 6 years younger. These are critical cells for learning and memory plasticity.
So it's really targeting the core machinery of memory.
Right. Seems like it. Plus, they saw reductions in things like oxidative damage markers and those protein clumps like A and p-tau.
Which are linked to conditions like Alzheimer's.
Exactly, and less neuroinflammation in the hippocampus, too. So a really comprehensive brain benefit package.
Incredible. Okay, what about other aging aspects? Bones, reproductive health, things that really impact quality of life.
On bone health, yes, positive effects. They saw less age-related bone loss around the teeth, in the gums, and less breakdown of that spongy trabecular bone structure you find in vertebrae, ends of long bones.
Which relates to osteoporosis risk, right?
Directly.
Yeah.
So potential there for preventing fractures and maintaining skeletal health, big implications for independence as we age. And then the reproductive system results were, well, quite striking, maybe even unexpected in their magnitude.
How so?
The SRC treatment led to superior biological age reversal compared to the standard WTCs in reproductive organs. In males' prostate, seminal vesicles tests, it was about a 4-year reversal with SRCs versus 3.3 with WTCs. And they even saw better sperm production, spermatogenesis.
Wow. And for females?
Ovarian aging slowed significantly. The egg cells themselves, the oocytes, showed a biological age reduction of over 5 years with SRCs.
5 years younger for egg cells.
Compared to about 3.4 years with WTCs.
Yeah.
It really does open up questions about extending reproductive health span, maybe even fertility, in ways we hadn't fully considered.
That is truly remarkable. Unexpected, like you said. What about the blood? It connects everything, right? Must have shown changes, too.
Oh, absolutely. Clear signs of rejuvenation in the blood. SRCs were better than WTCs at reversing those age-related gene expression patterns in immune cells, specifically PBMCs.
So a younger acting immune system.
In essence, yes.
Yeah.
Less senescence, less chronic inflammation in those blood cells, and at the same time, an upregulation of genes involved in DNA repair and autophagy.
Autophagy, that's the cellular cleanup crew, right?
Exactly. Getting rid of junk, recycling components. So the blood environment itself became healthier, more robust.
It's just amazing how these cells put into the bloodstream can have such wide-ranging effects. Lungs, brain, bones, blood. How do they do that? Are they physically traveling everywhere? Or is it something else?
That's a great question about the mechanism. It seems they don't have to physically go everywhere. A lot of the therapeutic benefit comes from what we call paracrine actions.
Paracrine, meaning they release signals.
Precisely. They secrete beneficial molecules that influence other cells, near and far. And a key part of this seems to be tiny packages called exosomes.
Exosomes, like little messenger bubbles.
That's a perfect analogy. Tiny mail packages sent out by the cells. And the exosomes derived from these SRCs are packed with good stuff. Geroprotective compounds, enhanced antioxidants, anti-inflammatory molecules, rejuvenating metabolites like spermine.
Spermine. Interesting name.
Yeah, it's a molecule known to be important for cellular health and longevity. So these SRC exosomes are loaded with it.
So it might not even be the cells themselves directly doing everything, but these packages they release.
That seems to be a major part of the story.
Did they test that idea? Like just using the exosomes?
They did, which really strengthens the case. In separate mouse studies, just injecting the SRC-derived exosomes without the cells showed significant anti-aging effects across multiple tissues: liver, lung, kidney, muscle, even brain.
Just the mail packages worked.
Seems so. And also, when they tested these exosomes on human cells in vitro, in lab dishes, neurons, ovarian cells, blood vessel cells, liver cells, the exosomes boosted vitality, cut senescence, increased proliferation, and reduced inflammation.
Wow. So maybe future therapies could be cell-free, just using these exosomes.
That's a very exciting possibility, yes. Could potentially be easier to produce, store, and administer. Has significant implications.
This is all incredibly promising, but, we have to ask, what are the limitations here? What's next?
Absolutely. It's crucial to be realistic. This is still initial evidence. Even though it's in primates, which is a big step, we need much longer, larger trials, especially in humans eventually, to see if these effects translate and persist long-term.
Right. Like, does the improved fertility actually lead to successful pregnancies long after treatment? Do cognitive benefits last for years?
Exactly. Those clinically relevant functional outcomes need confirmation. And while exosomes are clearly important, we need to fully understand all the mechanisms, other secreted factors, direct cell interactions. It's likely complex.
But the groundwork, it's definitely laid, isn't it?
Oh, absolutely. This research fundamentally shifts the conversation. It points towards a future where we might not just treat individual age-related diseases one by one, but develop potentially universal, maybe even off-the-shelf, human stem cell products or their derivatives like exosomes for a truly holistic intervention against the aging process itself.
Addressing the root causes, not just the symptoms.
That's the dream. Imagine managing aging comprehensively.
Okay, so let's try and wrap this up. What's the big picture takeaway from this deep dive? It seems these genetically engineered human stem cells, these SRCs, they can genuinely reduce multiple aging markers in primates significantly, improving tissue health, cognitive function across so many different organs.
That's the core finding. Remarkable biological age reversal and functional improvements.
And it's not just about living longer, is it? It's about extending health span. More years spent healthy and functional.
Exactly. Quality of life is key.
It's a really compelling glimpse into a future where maybe, just maybe, age-related decline could be dramatically slowed down or even reversed for you. It makes you think, doesn't it? If our cells can be nudged towards resilience, towards rejuvenation, what does that truly mean for how we view aging and our potential?
It's a profound question to ponder.
Well, on that thought, we really want to thank RevGenetics again. Their support for public science education is fantastic, and it makes deep dives like this possible. If you want to learn more about their commitment to science-backed health info or sign up for updates on research like this, just visit RevGenetics.com. We hope you enjoyed digging into this fascinating study with us today, and we look forward to exploring more cutting-edge science with you in the next deep dive.